Ashish, can you go ahead and share your screen, please?
Great, yeah, happy to.
Thank you. Ashish Manglik is an assistant professor in the departments of pharmaceutical chemistry and anesthesia and perioperative care here at UCSF. And he's our last basic science speaker for this part of the afternoon. And his title is Traversing Chemical Space to Elucidate GPCR Function.
Thanks, Alyssa, and thanks, Mark and Howard, for the invitation to come talk in this forum. So, before I get started, I must fully admit that my understanding of pain biology is incredibly rudimentary. And almost everything I've learned about pain, I've learned from Alan. So if I have something really wrong, it's probably Alan's fault. In fact, I want to point out that when I was a medical student at Stanford, almost, let's say 12 years ago now, in the neurobiology course, they couldn't find a pain person to come talk to us. So they actually got Alan to come talk to us. And what I've just put up here are some of my notes from my time at Stanford, from lectures that Alan gave, where you can see that the slides that he presents today on the right are still the same slides that he presented, almost 15 or so years ago. So again, I'm excited to be here. I'm still learning a lot about pain, and really our entry into pain biology comes from a deep interest in receptors and receptor proteins themselves. So my lab and my interest is primarily in how cells sense and respond to their outside world. And the primary way in which cells do this are by this fascinating set of receptors called G-protein coupled receptors. They're the largest family of proteins in the human genome. And they enable your cells to sense everything from hormones, proteins, ions, pH, light, or even force itself. And activation of this common fold of receptors by these incredibly diverse stimuli enables cells to sense these stimuli and then respond with a whole host of signaling cascades, everything from changes in second messengers like cyclic AMP or calcium to entire kinase pathways, most prominently the MAP kinase pathway. So it turns out that GPCRs control almost every aspect of your physiology, and that's why they're such perennially great drug targets. So everything from how you think, smell, taste, or feel to how you form social bonds with your kids, all of this in some way is modulated by a GPCR. And as a result, it's no surprise then that because these are such great professional sensors, they're also incredibly implicated not only in how we perceive pain, but also how we perceive our experience of pain, and then how that relates to the ability, for example, to find certain medications rewarding or not rewarding. Now, this is a great image from a recent review by, or not so recent, a review by Nigel Bunnett's group about 5 years ago, that just starts to get into the importance of G-protein coupled receptors, especially in pain, but similarly important in addiction. And what you can see in this diagram, this is kind of the general thing that you've already seen a lot of in a set of talks previously. Some kind of noxious stimulus being transmitted up, the so-called pain pathways. But a lot of the modulators of this perception are G-protein coupled receptors. And these are receptors that are all across a whole host of different families. So everything from receptors that recognize specific metabolites to receptors that have evolved to basically tune up or down that perception or that initial stimulus. So my lab is really interested again in the core logic of how G-protein coupled receptors work. In order to get at this core logic of how this large family of receptors work, we primarily do a lot of protein science, so a lot of biochemistry and biophysics. And we have two major goals. One is to understand how these receptors work at the atomic level, really at the most nitty-gritty level. And their long-term perspective is if we can understand these receptors and their most fundamental principles, then we can eventually hopefully use that kind of information to control signaling in very precise manners. And the second big goal is to actually use protein science itself to discover new pathways and eventually new biology. So I won't have that as much time to talk about the second side of things, mostly because these are very new projects in the lab. But I'll tell you a little bit more about our efforts to understand how GPCRs, and specifically GPCRs important in pain, work, and hopefully how that might lead to the ability to control signaling very precisely for pain therapeutics. Now, one of the big focuses of my work in the past and our work going forward in my independent lab here at UCSF is to understand opiate receptor signaling. And this is probably reviewed to the majority of this audience, but I'll spend just a second on it. So like many other G protein coupled receptors, the receptors for drugs like morphine or codeine are reside at the cell surface in a variety of different places in your body. And activation of these receptors enables them to engage with heterotrimeric G proteins. For the opiate receptor, the primary G protein is the inhibitory subtype, and that G protein then goes on to do a variety of things inside the cell. Now, classically, these activated receptors can engage molecules called beta arrestins, named because they arrest or turn off signaling. And one of the primary ways that they do this is by internalizing these receptors and preventing their signaling from the plasma membrane. Now, the combined output of all of this is, of course, the beneficial effects of any kind of opiate medication, as well as the side effects. But what's become clear over the past couple of decades is that these kind of textbook level examples of GPCR signaling are probably simplistic, and there's incredible complexity in GPCR signaling. So what drives that complexity? And that's something I'll preview a little bit of here. For any given GPCR, we've known for decades that we can turn them on or off with drugs. For opiate receptors, for example, this is probably one of the poster childs, in addition to adrenergic receptors, for how drugs can turn receptors on or off. But what's become clear in the past couple of decades is that even on top of drugs that turn receptors on or off, we can often find other molecules that can tune the level of signaling, so-called allosteric modulators. And this kind of pharmacology has led to significant excitement for a whole host of receptors. On top of that, we know that for certain receptors, two drugs acting at the same receptor can have very different kinds of signaling outputs. Alan alluded to the notion of bias signaling for opiate receptors, but we know that as a class for all GPCRs, or for many GPCRs, there are examples where we can find either endogenous hormones or exogenous small molecule modulators that could bias signaling from the same receptor down one pathway among many. For some GPCRs, there's evidence that receptors can find a partner to dance with, and depending on the partner that they dance with, the signaling outcome can change. We know that many GPCRs are constitutively active, so that even in the absence of some sort of stimulus, their activity, that they're active. And in the brain, for a number of orphan G protein coupled receptors, this is probably quite important for how they work. And then finally, really work by Mark von Zastra here at UCSF and Nigel Bunnett, who we heard from in the PARC lecture series just last week or the week before. What started to become clear is that even where receptors signal in a cell is really critical to the pathways they activate. So for example, receptors that activate signaling pathways from the plasma membrane can have one set of consequences, whereas receptors that are internalized by arrestins, for example, and continue to signal from the endosomes can have completely different signaling pathways. So it's really this complexity that we're after, and we're hoping to take these little cartoons that I've drawn for you and in a long-term perspective, understand these kinds of complexity from the atomic perspective. So really, if the past 5 or 6 decades has been to understand pharmacology from the perspective of cells, what we hope to provide on top of that is really atomic level insights into what drives pharmacology. And the phrase that I like to use sometimes is the notion of structural pharmacology. Now, if we want to understand proteins at this level of detail, there's a variety of methods. And excitingly, our ability to see proteins at the most nitty-gritty level has completely been revolutionized over the past decade. Again, some technologies that were developed here at UCSF. Classically, in order to see proteins themselves, the most high yield method was X-ray crystallography. And the idea here is that if we can coax proteins to form a very repeating array, very similar to how sodium chloride forms the nice little crystals of salt in your shaker. And if we can shoot those with X-rays, we can understand where the electrons are in space and and as a result, figure out what the three-dimensional structures of proteins look like. And the big challenge with X-ray crystallography is just the ability to coax proteins into crystals in the first place. By contrast, what's happened over the past decade is approaches in electron microscopy, specifically cryogenic electron microscopy, where we directly freeze protein samples in what we call vitreous ice, enables us to image them with electron beams instead. And we can take these very low signal to noise images and average many, many particles together to eventually get high resolution reconstruction. So again, some of this technology to enable this was developed here at UCSF, but primarily now with this second method, we're able to get really beautiful pictures of really not only how proteins look, but perhaps even how they move to achieve their functions. So with primarily by X-ray crystallography, you know, in the early part of this decade, previous decade, we were able to solve numbers of crystal structures of opiate receptors, some with work that I was involved with and other work by many other groups across the world. But for opiate receptors in particular, we have an incredibly rich perspective on how these receptors actually recognize their hormones or recognize exogenous drugs, and how they get turned on or turned off by drugs. So these are just a panoply of structures of various opiate receptors in the off state on the left here, or in the on state. And with these structures, we've been able to, with Brian Schweikit's lab here at UCSF, use computational docking approaches to find new molecules that have never existed before that target these receptors. So I'll very briefly just go over this quick example of using docking to find a new molecule, which we call PZM21, which Alan alluded to earlier. So I don't want to spend a ton of time on this molecule. But this molecule was different from known opiates in the sense that it fulfilled some of the criteria of so-called being a biased agonist of opiate receptors. But what's exciting about this is not specifically that this molecule has, you know, necessarily beneficial effects or increased therapeutic index on opiates, but just the fact that we can use this engine to rapidly discover molecules that have very differentiated kinds of activity at a given GPCR. And if you think about the ability to find molecules in this manner, it's a very exciting place to be. So again, that was kind of our first foray into just using structures to find molecules. What we're doing now is using the whole panoply of opiate receptor structures to find very bespoke or precision pharmacology within this subfamily. So for some of the people that are familiar with opiate pharmacology, one of the most interesting compounds that's been discovered for opiates for many decades is this molecule buprenorphine. And one of the fascinating things about buprenorphine is that while it's a partial agonist, the mu opioid receptor, it has antagonist properties to the kappa opioid receptor, and at least some of the clinical experience suggests that it has a relative ceiling effect for respiratory suppression. So with the structures of opiate receptors now, for example, the kappa receptor both in the on and the off state, and the opiate receptor in the on and the off state, what we can start to do is take combined data sets. So for example, let's say we wanted a molecule like buprenorphine that would bind to the active opiate receptor, but not to the inactive opiate receptor. But similar to buprenorphine would be a kappa antagonist, so perhaps it would effectively engage the inactive state of the kappa opiate receptor, but not the active state. We can take those two conformations and do combinatorial docking. And here, we've been greatly enabled by the Schweikit's lab ability to screen enormously large areas of chemical space. So I'm just looking at the time and recognizing that I'm running out, or I'm already halfway through. So I'm just going to get to the punchline of this, which is that traditionally what we can look at by virtual screening approaches, or by any other approach in screening, are really a small, very, very, very small subset of chemical space. And there's a variety of people out there that have tried to estimate, if you could think of all drug-like molecules that exist, what would that space be? And the number that is often thrown around in this space is a number that's 10 to the 60th. That's a really large number. Just to give you some perspective, that number is log orders larger than all of the atoms in our solar system, far larger than the number of stars in the universe. And by contrast, the total number of approved drugs, or even what we can assay by high throughput screening methods, is far, far, far smaller. So really, if we want to find new chemical matter, the larger the libraries that we can screen, the more productive we're likely going to be. So the challenge for drug discovery, again, is we want to find these incredibly rare needles, and our haystacks aren't just, that one needle in this specific haystack or this set of haystacks, but really haystacks that are as big as the universe. So what's exciting about some of the new methods in Brian Schweikat's lab are the generation of these incredibly large screening libraries. And for opiate receptors, Chase Webb, a joint student between my lab and Brian's lab, has been applying these what Brian likes to call large-scale docking or LSD docking campaigns against opiate receptors. And what Chase is doing is exactly what I described earlier. He's using these super large libraries to look for molecules that might have the same kinds of pharmacology as, for example, a drug like buprenorphine. So what he's doing is finding molecules that are at the intersection of predicted binding to the kappa off state. So there would be presumably kappa antagonists, as well as molecules that fit simultaneously at the mu opioid receptor active state. And the hope is that if we can find completely new chemical matter in this space, we can have molecules that have the kind of unique properties of buprenorphine, but don't come with the liabilities. But again, this is just one example. The exciting thing that Chase is now also doing is combining this kind of differential pharmacology for a variety of the opiate receptors. And in broader efforts within Brian's lab, the hope is to pair this kinds of bespoke pharmacology, for example, for opiate receptors with, for example, alpha-2 receptors worked on by Alyssa Fink in his lab, or for any other sets of receptors that have been implicated in pain pharmacology. So I'll stop there for opiate receptors. I'll tell you in the next, about 10 or so minutes, our work on a completely different receptor system. And this is a system where, similar to opiate receptors, there's been a lot of excitement and a lot of tears from the perspective of drug development. So the other system that we've been focusing on a lot in my lab right now is the Neurochinan receptor. And many of you, will probably recognize this receptor as a storied failure in the development of pain therapeutics. Just as a quick reminder for the role of neurokinin signaling in pain perception, one of the key things that neurokinin receptors do is they're co-released with glutamate and with CGRP, and are responsible for some of the perception of pain. So this is a nice paper from Allen's lab from the late 90s, where they show that that mutation of either the peptide, in this case neurokinin or substance P, or the receptor completely ablates pain perception in at least rodents. So there's a lot of interest in the late 90s and early 2000s to develop antagonists for the neurokinin receptor for pain. Unfortunately, almost all of these molecules failed incredibly miserably in clinical trials. So for those of you who were at Nigel Bunnett's talk last week, you probably heard a little bit about some of the ideas that they have on why some of these failures might have happened, and their proposition is that this relates again to the fact that these receptors perhaps could signal through endosomes. But what's really intriguing about the neurokinin receptor is that it displays a lot of these different kinds of complexity. So Nigel talked to you a little bit about the notion of endosomal signaling, but there's clear examples for the neurokinin receptor that it, for example, has different levels of constitutive signaling. What I'll talk to you a little bit more about today, that it has some examples of biased signaling, and there are described allosteric modulators for the neurokinin receptor that can, again, switch signaling specificity among different pathways. And one of the things that we, like broadly in the GPCR field now, have started to believe is that the reason why these receptors have such immense signaling complexity in part reflects their remarkable protein complexity. So, the textbook example or the textbook logic that we like to give for GPCRs is that these receptors are kind of bimodal switches. They turn on and they do a bunch of stuff. But increasingly, our view of these receptors is that once they become activated by some sort of hormone or drug, that their energy landscapes are really quite flat, and as a result, they exist in a huge plethora of conformations. And this conformational complexity is what gives rise to this enormous signaling complexity, and that eventually is what gives rise to the complexity and behavior that we observed for a whole host of GPCR signaling pathways. So for neurokinin receptors, it's been known for some time that there's a pairing of endogenous neuropeptides that target neurokinin receptor 1 and related analogs. So turns out that there are 3 gene products that make tachykinins or neurokinins. The one that I'll tell you about a little bit more about today is the outcome of the TAC1 gene, which makes both substance P and a related neuropeptide called neurokinin A. And these set of neuropeptides hit 3 different neurokinin receptors, neurokinin 1, 2, and 3 receptors, each again made by a different gene product. Now, initially, it was thought that substance P was primarily specific for neurokinin 1, whereas neurokinin A was specific for a different neurokinin receptor, neurokinin 2. But what's become clear, again, became clear almost 2 decades ago, is that neurokinin A actually has quite a bit of activity at the neurokinin 1 receptor. And this is, again, a paper from Alan's lab from the late 90s showing in vivo that both substance P and neurokinin A can act through the neurokinin 1 receptor to drive its internalization in neurons. What's interesting about neurokinin A and substance P is that they have differential signaling outcomes. So the molecular pharmacology of these receptors shows that substance P can both activate the GS pathway or the GQ pathway, leading to 2 very different downstream second messengers, either cyclic AMP or calcium. By contrast, neurokinin A is selective for the GQ pathway and actually doesn't cause any amount of cyclic AMP signaling. And there have been a lot of work to show this kind of activity, again, in the late 90s. And on top of that, a number of groups had identified a number of either mutations or other ways to modulate neurokinin receptor signaling, neurokinin 1 receptor signaling, to again give similar phenotypes. The outcome of a lot of this work was the proposal that this receptor exists in at least 2 conformations that perhaps gives rise to this complexity, the complexity of this kind of behavior. So we've again been very interested in this receptor, really from the perspective of trying to understand whether differential conformations can give rise to differential signaling outcomes. And in order to get at this, a couple of students in my lab, Julian Harris and Brian Faust, Brian is a joint student with Yifan Cheng's lab, have been trying to get at trying to understand neurokinin 1 receptor signaling by electron microscopy. So very recently, they determined a structure of the NK1R receptor, which is again here shown in purple, the various G protein components on the bottom. And for the first time, they've been able to map the binding of substance P to the neurokinin 1 receptor. And that's basically shown in orange here. Again, the receptor has changed color here to green, and the G protein, just at least a part of the G protein, is in yellow. Zooming in, we can start to explain some of the interesting observations that have been made in the past. For example, again, here's an overlay. You were comparing the binding of the antagonist, or one of the clinically used antagonists, nitipitant in yellow, with substance P in orange. And what's really remarkable about this is that the activator and the antagonist largely occupy 2 very different pockets. So that's kind of neat. But really, what we're more excited about, and again, this is very preliminary, so we're just starting to get hints on this, is again this notion of conformational heterogeneity. So what Brian has started to pull out from some of this work, I'll just show this movie here, is perhaps evidence of multiple conformations of the neurokinin receptor associated with perhaps differential signaling outcomes. So what you're looking at here is a morph between 2 electron density maps that come out of our cryo-EM data, where state A is in this blue color, so that's basically this map right here in blue, and then it'll morph to the yellow state, which is we think an alternative state. And this is now perhaps experimental data that's again suggesting directly that this receptor exists in multiple conformations. And what we're hoping to do now is really correlate that to the unique signaling outcomes associated with these 2 different peptides. Okay, so what does this practically mean for pain drug discovery? I think we're at a very exciting time where we know an enormous amount about the complexity of neuropeptides and how they act, or how drugs act. We can increasingly use structural biology methods combined with the kinds of approaches that Brian's lab has pioneered and developed over the past 20 years to rapidly find molecules. And the exciting thing about GPCRs is that any two molecules acting at the same receptor are likely going to have very different or very unique outcomes just because these receptors are incredibly conformationally complex. And in order to kind of leverage some of this work, we've been engaging in an exciting approach to do this, funded by DARPA, a project led by Brian called the Panacea Program, ambitiously called the Panacea Program, with a whole host of collaborators here at UCSF and across, in the Ukraine, as well as at UNC, to use these kinds of approaches to rapidly discover new molecules against known pain targets, with Alan's lab and Nevin's lab discover new pain targets and apply similar approaches. But really at the heart of the kind of work that we do, we're very interested in, once you have a target, understanding the conformational complexity that drives signaling complexity, and then with the hopes of finding new molecules, being able to push and pull on this complexity in prescribed manner. So that's kind of our long-term goal. So with that, I'll stop, take questions. I know I'm a little bit over time, but hopefully that gives you some overview of where we're hoping to interface with PARC and with pain and addiction pathways.
That's great, Ashish. Thank you so much. We will, I think, have a few minutes for questions just for Ashish, and then I'll open it up for all of our speakers for this part of the afternoon session. Let me start off by asking a little bit about the sort of energy landscape that you showed. And can you give us an idea of what kind of timescales you're thinking about when you say these proteins wiggle around, and how they move from one somewhat stable conformation to another?
Yeah, great question, Alyssa. So, typically when we think about protein motions, these are all very fast. We're talking, microseconds or millisecond kind of motions. But what's really neat about the neurokinin receptor is that the old school pharmacology, this is now decades old pharmacology, suggests that these two conformations, exist in cells, when you're doing your old school radioligand binding experiments. So those are now two conformations that are switching on some very, very, very slow time scale. So again, for the kinds of motions that I showed in those little movies, or that we're trying to get at by EM, those are probably very fast motions. But some of these things could be very slow, slow changes that really reflect the amount of a given receptor, quote unquote, binding site in the old pharmacology parlance, in tissues.
Mm-hmm. I think Alan was waiting for a question.
Sorry, Mark here, I have to interrupt. Ashish, thanks so much. Can you stop sharing your screen? And I just need to put up, thank you, 1 second, just as a housekeeping part of this. So we have the, sorry to interrupt, but this QR code will help us for those that have been participating. If you just take a moment as we're discussing to also start completing, or in the future complete a survey for the retreat, it'd be hugely helpful in the future. Thank you.
So
sorry. Go ahead, Ashish. Sorry.
Yeah. So Ashish, thanks. I guess there's sort of two related questions. The big problem as a preclinical basic scientist, and you're aware of this, is that the fact is the antagonists work in the animals for pain, and they clearly failed miserably in humans. And that's always a worrisome problem, the translation question. But the one interesting thing, and I don't know if structurally we know anything about it, is my recollection is when Merck was doing all this work, that the hamster is a better predictor of what happens in the human than the rat or the mouse. And you get this, I think, thumping behavior that is blocked by the antagonist. Is there anything known about the structure of the human receptor versus the rat that would suggest that there are really significant functional differences?
So we've looked at the sequences themselves. Obviously we focused on the human receptor primarily for all of our structural work. And there doesn't seem to be something that just pops out as a clear difference that might drive, lack of binding or something like that. or even between the hamster and the human. So at this point, I don't think we have a great grapple on that. I think one thing that I found intriguing, and I think this is where, you know, Alan, this is really harkening back to your work in this space, and is the notion that certain antagonists are better at antagonizing, for example, neurokinin A signaling in the cord versus, you know, substance P signaling. And I think the level of complexity there might be more so than simple, you know, this thing binds and it binds tightly. The actual amount of functional antagonism of substance P, you know, bound to its receptor may not just reflect whatever the in vitro pharmacology was, right? So that's, again, a very high level take on it. My perspective on this is I think we need to understand more about what these two different peptides do from both the, you know, very molecular pharmacology or even, you know, reductionist structural pharmacology approaches, and then eventually more about that from the perspective of from actual, you know, physiology. I think, you know, people had started to do that, but there's really not a lot known about what the role of neurokinin A is in a variety of neurokinin receptor biology.
Dorit, did you have a question?
Yes, is there time?
Yeah, go ahead. We're into kind of back and forth now, so please.
Okay, so great presentation and work. What I don't understand, and I'm sure you have an answer for that, when you activate the GPCR, whether it's GQ, GI, GS, the end result is the same. So, I mean, it's the same, meaning, you know, doesn't matter how, you know, what is the conformation of change, how long the receptor is activated, etc. You know, you have kinase signaling, you have PKA signaling going up, going down, you have PLC signaling, calcium release. How do you envision getting, you know, drugs targeting these receptors where the signaling end result is dictated by something else? Do you know what I mean?
So I'd beg to differ that the outcome, you know, is similar, right? I think that there are clear examples even within opiates that, let's say, a partial agonist has a different, you know, outcome than a full agonist, right? So I think, and certainly for receptors for which there are, you know, ways to bias signaling down one pathway versus another, there are behavioral outcomes, and we can, you know, find examples of those. I don't think that, you know, a GI coupled, let's say, even if we just consider a GI coupled receptor or a GS coupled receptor acting in a given tissue, that, you know, their outcomes are the same, right? I mean, they're fundamentally different signaling outcomes, even if they impinge upon some downstream, you know, pathway like MAP kinase in a similar manner, right? Their upstream outcomes are really quite different, and that can have pretty profound differences. So now, whether we can prospectively, you know, connect the dots between a signaling bias level or you know, a preferential signaling level at the level of, you know, calcium at GQ or something, and behavior, I think we're very far away from being able to do that prospectively. But I think from the molecular pharmacology perspective, or, you know, some sort of response at a very proximal level, I think they're pretty good examples of that kind of behavior.
Thank you.
I'll read a question from the Q&A from Arthur Wood. Can a better understanding of mu opioid GPCR help with discovery of agonist drugs that don't lead to tolerance? Obviously, that's a problem with chronic opioid use. Big question.
Yeah, I mean, this is like the holy grail question, right? And I think there's a lot of thought in this space about what the role of either biased agonists could be, or perhaps molecules that are low efficacy partial agonists. So things that are maybe have just enough tone to provide some amount of pain relief, but lead to lower amounts of tolerance. Or alternatively, molecules that combine opiate receptor tone with some other, either opiate receptor like the kappa receptor or some other GPCR to, decrease the total amount of tolerance. So, from the perspective of what we can do from these very nitty-gritty approaches, I think the structural biology helps us crank the Schweikert lab engine pretty rapidly to find new molecules. But again, I think we're pretty, you still have to have a pretty clear hypothesis of what you think a way to decrease tolerance is and really test that.
Let me ask you a slightly different question, which is, there's some evidence in rodent functional studies at sort of the individual neuron level that say in some neurons you get GI coupling of mu-opioid receptors. In other cases, you get GO coupling. The function is maybe a little bit different depending on whether you look at somatic function versus dendritic or terminal function. What is your take on how you go from that crystal structure to what part of the neuron or which instantiation of the receptor complex you can target?
Yeah, so I think it's hard for, to separate out things like GI versus GO coupling. I mean, by and large, from a structural perspective, they're pretty similar. So, but, I think for something like the Neurochinan receptor, that becomes far more interesting. If you have, if you, for example, knew the sites of release of substance P and or Neurokinin A, and then also knew on the receiving end, whether there's GS or GQ there, that really tunes your idea about what the outcome of that event is versus, simplistically just, one peptide, one receptor, one signaling outcome. Yep.
Can I ask a general question? I know plenty of individuals who don't believe tolerance is a major clinical problem. What is the sense around this table?
It's a interesting question. I would have to say it's a huge problem, and at least when patients come into the perioperative or come in for hospitalization, and they are, we present to general services and they're completely, you call the term resistant to what we would think of as typical doses, or doses that would normally produce an analgesic or endpoint effect within a dose range. And then very rapidly, there's dose escalation with life-threatening side effect profiles, the concept that it's somehow protective up to a point, and no one knows where that is from person to person in terms of respiratory depression or the other side effects. So at least in the acute care setting, it turns into be a huge challenge for most clinicians.
But I'm talking more about not the acute setting. Mm-hmm. Very different. So the patient who's taking opiates for back pain, some of them escalate a lot, and my understanding is others don't. Some clinicians think the drug doesn't do a damn bit of good for them. Others think it's effective. And that dichotomy of opinion clinically is really kind of frightening.
I can tell you from my own experiences, a lot of patients and opioids that you're exactly correct. What's interesting is that very often patients will do well, with a set dose of an opioid for months. And then something happens and they need to begin to escalate the dose a little bit. Sometimes that can be very, very slow. I think that whenever that does happen, it's time to reevaluate the patient to make sure that there hasn't been progression of the underlying disease, or that there's some kind of opioid use disorder going on. Either one is possible because a use disorder can develop late in the clinical course of a patient with chronic pain. It's not always rapid.
I'd like to make a couple of comments here, if I might. One is that Pam Palmer, about 15, 20 years ago, published a series of papers in both humans and in rats, showing that tolerance development was very rapid in the young rat and young human, and that over the age of 65, the rate of tolerance slope was extremely flat. I know clinically speaking, I'm almost always more nervous about opioids in the young. And so this actually leads me to a question to Akash and to Stefan about Stefan's statements about pediatric usage of opioids. I have seen, undoubtedly, I've seen tolerance develop in the very, very young, having dealt with cancer patients, osteosarcomas in particular in the pediatric population, on doses that would frighten even the most hardy opioid use disorder heroin addict. And so I've seen 500 to 1,000 milligrams a day for a 12-year-old who is not getting adequate analgesic responses. So my question to Stefan and Akish and Howard and the like is that I certainly have seen this. It's certainly there. It's certainly real. My dilemma and my concern, and especially by the time I get called to see these folks, is that am I working with Robert Goldsby in the pediatric oncology field, says that about half of these patients can be successfully treated for their cancer and they'll go on to be in remission. But if they're on high dose opiates from 12 to 17 on a 500 to 1,000 a day for 5 years, what is the likelihood, Ashish and others, that beta-arrestin has reverted back to a normal state when they're 25 or 30? I'm very concerned about this extremely high dose. I definitely want to be able to treat severely cancer kids, but it's just a concern that I have, and I don't know anyone that can tell me about the long-term consequences, not of the acute examples that Stefan gave earlier of acute surgery, but after 5 years of high-dose opioids, all of a sudden to come to an end to that, how much longer do you think we need to wait, if at all, that it'll come back to normal? When we see the elderly not develop tolerance, but that's assumed that they will never get back to where they were when they were 20 years old. So I'm a little curious about that, and I'd like anybody's thoughts on it.
I can start, or I can wait. Who's moderating?
Go for it.
So I think you're right. We do know that the flip side of neuroplasticity is a higher degree of tolerance. So the younger child is, the younger brain is, the faster it will become tolerant to an opioid. So if we compare premature infants to neonates, to 1-year-olds, to toddlers, to children, and start a fentanyl continuous infusion, the younger the brain, the flip side of neuroplasticity is that it will become tolerant. So that is an issue. We do know that any given opioid in pediatrics, and I think the same is true for adults, only is going to provide excellent analgesia for acute pain in about 40, maybe 50% of the patient. In other words, if we have one opioid and we're giving it, half of them will not have good analgesia, and that's the purpose of opioid rotation. So, so I think the dark secret of pediatric pain medicine, especially in adult as well, I think that is a ceiling effect for certain drugs. So this may be in part of development of tolerance. It may be in part of that medications such as morphine and hydromorphone are metabolized into bad guys, morphine 3-glucuronide, into hydromorphone 3-glucuronide, which then accumulate. And if we have kids on that like 50 times the standard dose, 100 times the standard dose, this stuff is probably not working. And this is the wrong drug. And either, if opiates are right, you want to think about methadone, or another sort of multimodal analgesic, or the whole. It's hard to imagine why a cancer patient would be, a pediatric cancer patient would be in opiates for 5 years. Yeah, so that is probably something we would like to see sort of in the pain service. We often, unless it's really sort of end of life, would be 2-3 bone marrow transplant, but that is really not what we're seeing. We should be able to get them off opioids using multimodal analgesia. often those high doses are happening in patients who do not have access to psychology, who do not have access to physical therapy, they don't use sort of adjuvants, and somebody may just sort of keep prescribing extremely high doses of opioids. And outside the palliative care realm, I think we found this to be less and less helpful.
And do you think that once they are off opiates, they've been on high dose for 3 to 5 years, that they're likely to return to normal pharmacological responses, 4 or 5
I'm
years?
not aware of good data. I certainly have seen many patients with who get cardiac surgery and have their sort of palliative surgeries sort of as an infant, and then as a 1-year-old, and then sort of a few years later down. And it seems that when they come back 3 or 4 years later, they certainly do require higher opioid doses than an otherwise opioid-naive child. So I'm not aware that there's good data this, but in clinical experience, we certainly see kids being tolerant, so I don't think that they go back to like sort of zero receptor-wise like an opioid-naive patient.
So Akish, would it be consistent with your thoughts on how gene-coupled proteins receptors are expressed, and maybe
I must admit, I know absolutely nothing about the evolving mind, from the development and how any of this coupling, from the atomic perspective changes. So I fascinating discussion from Stefan, but I have basically nothing to add.
Okay, thanks.
Can I ask a question, or are we out of time?
No, I think maybe one more question, then we're going to turn it over to Howard.
Who's moderating?
PARC's moderating.
Sorry, sorry, and I don't mean to grab it from Alyssa, but I think we're going to switch it over to Howard at some point, and so that's what maybe next steps are.
Okay. Well, I'm ready.
Okay.
I think we have time for Stefan's question, please.
Yeah, Stefan, we're not going anywhere.
Yeah, please go ahead.
So so for the clinician, right, going asking for the basic scientist here, Ashish and Al, so my concept is a mu opioid, morphine, fentanyl, oxycodone, hydromorphone, binds to the mu receptor. It uncouples G protein. And usually the way I teach it is that there's like a bad G protein and a good G protein. So we have the inhibitory G protein, which causes much of the analgesic effect. We have the bad G protein, the stimulatory G protein, which causes lots of the side effects, such as constipation. And the good news about the bad G protein is that it's far more sensitive to naloxone. So that explains why giving low-dose naloxone at a whiff of a splash at 0.5 to 1 to 2 mics per kilogram an hour seems to be helpful for itching or urinary retention or nausea and vomiting and constipation. So my question then is, where do the beta-arrestins come in? Are they part of the stimulatory G proteins, or is this a completely different molecule? And then I just have to get my side effect profiles caused by stimulatory G proteins and beta-arrestins right.
The answer that I'll give is going to be different from Ashish. I'm going to let him go first.
So, there's relatively little evidence, at least that I know of, that the primary side effects of opiates are driven by anything besides GI coupling. So really, even the primary side effects of respiratory suppression or constipation are really all driven through GI or GO coupling, and really being able to resolve out, the GO or GI component is a little bit challenging right now. But really, to me at least, the primary mechanism of action of basically all opiates is, binding to the opiate receptor and then release of the inhibitory subtype G protein. And naloxone basically functions as a competitive antagonist and prevents or blocks binding of, either endogenous opiates or exogenous opiates. Probably the effectiveness of the little whiff of naloxone may just come from the fact that some of the side effects, the pruritogenic side effects, may require a dose response that is a little bit higher, requires a little bit higher doses than perhaps the analgesic effect, and you can tune out that some of that in some ways, and that's probably part of the reason why some of the partial agonists like buprenorphine gain some of their therapeutic index, is that they're they provide a little bit more tone on some of the analgesic effects, and that tone is not sufficient to meet the higher tone required for some of the side effects. But that's again my thinking on it. The arrestin molecule, again, classically is a completely different molecule and was thought to turn off signaling. So it basically is responsible for pulling receptors out of the membrane as one of the steps. But what has become clear is that these receptors that are not in the membrane or on the plasma membrane anymore still continue to do interesting things, and that's some of the work that we're doing with Mark Lanzastro's lab here, is that those internalized receptors can go on to do their own thing. With Mark's lab, we've shown that that those internalized opiate receptors can still get activated by opiates, specifically small molecule opiates like morphine, and that might have important consequences.
So just a briefie, do you think there's more than one form of the mu receptor?
It's a long discussion.
Amino acids?
A very long
yes
discussion.
Or no? Yes or no?
Maybe.
Okay.
Yeah.
That's good enough. I'm just trying to get some laughs here.
In fact, Ashish and I are on the same page on that question. Stefan, much of what, no, I'm talking about the first question.
Okay.
The issue of the excitatory effect, that story is based on absolutely reproducible results. There was even Pain Pharmaceuticals, if you recall, was a company developed based on Stanley Crane's work that there's a subtle excitatory effect of opiates at low dose. That has never been reproduced, and the company went nowhere. And low dose naloxone combinations with morphine never did anything, did not go anywhere. I think in the case, a good example that suggests that Ashish is right about the dose issue is, of course, as you know better than I, one of the best ways to get opioid-induced itch is to give it spinally. And then by giving it spinally, you get both itch and pain relief, and then you give a low dose of systemic opiate. And the idea of systemic naloxone, the idea is that you can pick out the itch component, but save the analgesic component. I think it's a circuit question, that what drives the itch versus the pain, but it's all through the same receptor, and it's inhibitory.
Well, I'm just going to jump in there for a second, because Howard and I did publish a paper a few years ago showing that at least in some neurons in the VTA, you can get a direct excitatory effect of activating the mu opioid receptor. It was a minority of neurons, but it was there. It was blocked by a selective mu opioid receptor antagonist.
Doesn't work in the knockout mouse?
We were in a rat. We didn't do it in a mouse, but it was completely blocked by CTAP. We got it in the same cell. We were able to subsequently block it with CTAP every time.
Okay.
And it was at a very low dose.
Yeah. So that effect was also, it was an equal nanomolar dose to the activation of GERK, but 2 orders of magnitude more sensitive to the inhibition of neurotransmitter release onto VTA neurons. So there is something, I think, a little bit more complicated going on there.
Thank you.
Okay, I think
Howard.
you are up, Howard.